Thank you for coming to this seminar series of Victimat. It's a great pleasure for me to introduce you to the speaker, which is Jordi Garcia-Ojalvo. Most of you or some of you know me because it's around in the field of biophysics, in the forefront of the field of biophysics in Spain for quite a while. So it's a great pleasure for me. I know him personally. He's a very good scientist, and I have to say, a very good person as well. So I hope you enjoy his talk. And also, I would like to say in the last, so we have a meeting of the biophysics of the Victimat related people like a couple of weeks ago or 2 weeks ago. And one of the questions that was during the discussions was like, how do we as physicists communicate with biologists? So how we bridge this gap in our language between physics and biology to establish a fruitful and long-term collaboration. And this is a difficult question also, but I think that one of the people that has this answer is Jordi, because he's been collaborating with biology for a very long time, and hopefully today we will show us some of the results. Also, one quick announcement for the students: if you need like a certificate, just talk with Salvatore after the end of the seminar. Okay. Okay. So nothing else. Thank you very much, Jordi, for.
Okay. Yes. So good morning, everyone. Thank you so much for coming. It's really I'm very happy to be here. As David says, I mean, I know him, many of you for many years. I consider you my friends, and I'm so happy. I mean, this is one of the nice things of this profession, right? That you have friends all over the place, and I don't know, this is so nice. So, thank you for being here. Some of you maybe are here as a master. They told me as a student of the master's degree. Hopefully, you are not forced to come. If you are forced to come, I apologize. But then I hope that I will somewhat convince you that this might be a little bit interesting. So I am a physicist by training, and I like very much what David said about how to communicate between disciplines, and this is one of the things. In fact, I have to say, well, first of all, let me tell you, I forgot to put the first slide. The second slide I would like to put here is the slide of my collaborator. To be honest with you, I just forgot it because I was doing this thing very quickly, and then I wanted to do it. And I have to tell you, it's very important because what I'm going to be explaining today is a genuine collaboration between physicists and biologists. And also it helps that you are friends. I'm going to show you mostly a lot of experiments, and this comes to the communications part, experiments that I have not done. So what we do is we do the theoretical and computational analysis in a very circular manner. So the experiments fit the theory, and the theory fits the experiments, which is not so common sometimes, but I think this is the goal. And you can do this if you have a friend and you're collaborating. In my case, the friend is Professor Goodall from the University of California, San Diego. You don't see here his name, and I will put him on the end, but I should have started with this. So we have been working together on this particular topic for 20 years in general, and in this particular topic for more than 10 years. And what I would like to do today is a little overview of things that we have been doing in this topic for all this time. So some of the things are going to be a bit old, and then I will build up towards some more recent stuff. I also remember that I gave a talk here 6 years ago or 7 years ago. And thankfully, those of you who are students, you were not here, so you will not see some repetition, but some of you might see something that you have already seen. I apologize for that. And going back to the communication part, what I have to tell you is that I always use the same slides. I don't know. So the same slides I always use when I talk to physicists or biologists. And sometimes, somehow they don't complain. I mean, maybe you will complain today. So I think it works somehow, and the way it works is that I don't enter too much into the details, which might be not too good for some of you, but that's the way I so I just wanted to tell you regarding this comment that David said about how to communicate with biologists. Well, I leave the slides not thinking of the audience. My my discourse is going to depend on the audience, but not the slides. So that's what I wanted to say first. So let me start. Okay, so as I was saying, I'm a physicist, and some of you, I'm not sure what is the fraction, are interested or working in biological problems, what we would call biophysics. Biophysics, by the way, is interested in the definition of the field, because I consider myself a biophysicist, but field of biophysics has a very specific meaning in some sense. But I think we consider ourselves as biophysicists, and those of you who are doing the masters. But anyway, I don't know how many of you are already working on this. I started working on this 20 years ago, after 10 years in which I was doing statistical physics. And I did it because really living systems are really amazing. No? I remember recently I heard, I don't know if you know David Krakauer. David Krakauer is the director of the Santa Fe Institute, and he defines living systems as problem-solving matter. I don't know if you heard that. So he defines living matter as problem-solving matter, which I think is interesting, right? So what separates living matter from non-living matter is the ability of living matter to adapt, to respond to the environment and to adapt in such a way that it enhances survival, which is not an easy task when you are competing with other types of problem-solving matter. And the result, the result of 3 billion years of evolution is what we have right now. Okay, so it is really amazing because we are witnessing an optimization, an optimized solution of a problem, which takes many different forms, and this is what we see. So what we do is, okay, we see what's going on, and then we have the advantage of seeing the results of this amazing process that has lasted for billions of years. So that is why I study living matter. Of course, one of the things that living matter has this is a positive and a negative things, and a negative aspect at the same time, because you have all these kinds of amazing behavior. Again, in terms of morphology, in terms of function, in terms of response to the environment. Okay, so you can have plants, you can have vertebrates developing in different ways, you can have slime molds navigating a complex landscape. So this is living matter. This is, if you want, you can call it problem-solving matter. But it is a fact that it's highly idiosyncratic. It's very, I mean, for a physicist, I don't know, sorry. Maybe, I'm not sure if you agree with me, but for a physicist, it's a bit unnerving that you have such large variety of things happening and such large variety of solutions. When we are doing a physics experiment, I don't know, I was talking with David a few minutes ago, 15 years ago and 20 years ago, I was working on laser physics. I was looking at chaotic lasers, and then you can set up your experiments, and your laser is not idiosyncratic. I mean, if you prepare the experiment in the same way, it's going to respond all the time in the same manner, more or less. You have variability, but that's fine. But then you enter into this field because you are attracted by the amazing behavior that this problem-solving matter has. And then the problem that we have as physicists is that I don't know how to handle this complexity, which results from the characteristics of the living matter. You have this. How can you find fundamental principles? I think that we as physicists, that's what we want to do. Try to find universal as possible, as universal as possible, and as fundamental as possible principles for this type of system. But it's very hard. I mean, I don't know. I have no solution. It's very hard. So, I mean, I don't know. Here I have another amazing example. Right? From a single cell, we emerge. And how does this happen? And of course, biologists have been working for decades in identifying all the details. We have a lot of information, a lot of variable, very, very information, and we try to understand how this happens. Now, what can we do as physicists? Well, one possibility is we try to find a model system. No? The idea is, okay, let's try to find a living system which has all these properties, but maybe it's a little bit less idiosyncratic, or you can control it better. And one of the main models, in my opinion, are bacteria. Okay, that's our bacteria. Bacteria are very simple cells. Some people call them bags of chemicals because they have basically no organelles. Everything is more or less well mixed. So they are relatively simple. you can perturb them a lot. I'm not sure. I don't think I'm going to enter a lot into these details, but you can generate mutants relatively easily, which means that the genetic background is different, and therefore the response is going to be different. And you can monitor them relatively nicely and simply. And this is what I would like to discuss with you. So these are very nice models. But you might say, oh, but this is too, well, one could consider this one could consider the hydrogen atom of biology. No, that's what I way I think about it. It's the simplest unit that I would like to understand. I know that some of you work molecularly, but I work, so this will be very simpler. But I want to consider a cell as a unit of such type of atom. Now, the problem is that maybe this is too simple. Because most of the examples I was telling you earlier, they're showing tissues. They're showing multiple cells working together that at the end of the day, they make us. So the question is, these unicellular organisms, how can they be informative? Maybe they are a hydrogen atom, but maybe this is telling us nothing. I would like to have some tissue, like our skin or like our muscles or like whatever. Can I have a tissue? Well, we can have a tissue from bacteria, and this is what I would like to discuss. Because bacteria, they can be considered units of living matter because they have collective behavior. They have a whole range of collective behavior. They form structures that can be unidimensional. So you have systems, and you have people here in Madrid that have been working on this, people like Saulares, have been working on this unidimensional living system and finding patterns and finding kinds of organization. For instance, very beautiful. So you have 1D systems, you have 2D systems, you have 3D systems with very different kinds. So you can build this kind of tissue, which is non-trivial, has non-trivial response, out of these relatively simple units, which are bacteria. So that's what I would like to discuss. This is what we have been working for a few years already, and I would like to give this overview now. So now I'm going to focus on this particular type of collective behavior with this particular type of bacterium. This is what's called a bacterial biofilm. Okay. This is a picture of a bacterial biofilm. Well, I know it looks like it lives on the sink in your bathroom. Just put your finger inside the sink in your bathroom and all the sticky thing. This is a bacterial biofilm. It's very hard to get rid of because this is super resistant for too many things. At the end of the day, what you have here is millions and millions of cells living together, stuck together. Because these cells, one of the things that they do is they secrete substances that make them stuck together. It's a cellular matrix, right? It's a cellular matrix that holds our skin together and our tissues together, okay? So these are cells embedded in this extracellular matrix, and this keeps them safe, and they keep against toxins and other kinds of stresses, okay? So they do this when they are stressed, okay? So when they are stressed, I'm not sure if there is I have any more now. Sorry. Sorry. I don't have it here. Maybe here. Here, you see. So the bacterium is floating, and at some point it attaches to a surface, and if they are stressed because they don't have enough food, then they start secreting this little substance, this ECM, it's a cellular matrix, and then they grow, and then they become large populations. Okay? This is a bacterial biofilm. So the first thing that we did many years ago was looking at the dynamics of formation of this type of structure. So to do that, we put all this bacteria on top of an agar sheet, which is food. It's basically water with some nutrients in a gel-like structure, and then you monitor it from below, and then you can get this. And then the idea is, how does this form? We did this quite a few years ago, but I wanted to show it to you because I thought that this was the right audience. So this is a film strip for snapshots in time. It's not very clear, but anyway, this was done by Munehiro and Sally. So this is from Grosswell's laboratory. So this is so you have an idea of the size, and then what you can see is that you form these little labyrinthic patterns. Okay, these little labyrinthic patterns. And what we wanted to understand is how this pattern form. It's just some kind of, I don't know, it's the tissue drying up. And it's just a physical result from the tissue drying up. It cannot be this because these things are alive. Okay, they are not dead. They are not just things that dry up. They are alive. We know that. Okay, so how they form? So then we were looking at this, and thanks to the optical processes, for instance, one of the things that we can do is we can use a fluorescent dye that stain cells that die. When the cell dies, it opens some holes and the dye enters and binds to the DNA. So when you see green, this means that the cell has died. So then you stain the entire biofilm, and then interestingly, you see these little patterns of cell death below these things that we call wrinkles. You have wrinkles into the structure, and then you have these little patterns of cell death. Okay. So there's certainly a correlation between cell death and the density of bacteria, if you will. Okay, so you have that. And then you have interesting patterns. You have interesting patterns, both early and late. I'm not going to go too much into the details. But now this, for a physicist, represents an interesting problem. So now if we forget about, oh, look, very interesting behavior of this system. Let me just show you. If you look at the dynamics of this thing as it's forming, in green you see the death. And as you can see here, you see a very interesting pattern of inverted spots. What caught my attention was that you have a field of cells, and then you have inverted spots. So if you like a pattern forming systems, some of you, it's very normal to have patterns of spots. These are anti-spots, because you have these holes here of living cells surrounded by this other cells. And eventually this doesn't disappear, it's just that it grows in the third dimension. But then the question is, how does this pattern arise? So then we as physicists, we said, okay, let's start to build a model. So we build a relatively simple but reaction diffusion system, which is continuous, in which we describe the local density of bacteria and the matrix that they are producing, the extracellular matrix. And then this is just how the local density is producing matrix, and then the matrix is restricting the expansion of the local density. You have stress, you have growth, so you can build this kind of reaction diffusion model. I am not going to go into this. I just wanted to show you the equations, no details. But then what we can do with a model like this is we can run simulations by using parameters which are educated guesses. I have to tell you, because many of these parameters we don't know the values, but you put educated guesses and then you let the system run. And then you check that the behavior is not sensitive to the parameters, okay? So that's what we can do at most, okay? So then we have this cell death pattern that is comparable to the one that we see in the experiments. And then what we can do is we can perturb the model. For instance, what happens if we remove the matrix component? Well, if we remove the matrix component from the system, we don't have a pattern anymore. So then what the model is telling you is that the production of the matrix is key. Because then this leads to this feedback, this positive feedback here, that eventually, together with this negative feedback and this positive feedback, is the one that is producing. So now the beautiful thing is that you can do this experimentally. So you have a model and you make a prediction and you say, look, if I prevent the cell from being able to produce matrix, I will have no pattern. So you do that. And you can generate a strain of a bacterium that is unable to produce the components of this extracellular matrix. And when you do it, you get, you can see this is one of the things that my colleague does in his laboratory. They can generate tons and tons of different kinds of mutants. And then you have all these mutants that affect different types of the different aspects of the bacterial metabolism and physiology. And the only one that really eliminates the pattern completely is the one that is associated with matrix production. Okay, so really this is an indication that maybe this is what's going on. And we as physicists, maybe what we can do is try to understand how things happen. Nowadays, I have to say, with AI, when some of you may think, some people are thinking that we shouldn't care. You just look for patterns using AI and we are done. I don't think that we as physicists should do that. We have to understand where things come from, and there are underlying mechanisms, so that's what we try to do. We hypothesize a mechanism, we build a model, we analyze it, and hopefully we validate it with experiments, and if this works, then we can consider that maybe this is what's happening. That's the story. By the way, this is only, if you will, correlation. Let me see if I have it here. We have a hypothesis underlying, and what is the mechanism of the formation of these wrinkles? Well, the mechanism would be that a cell death pattern forms, and then you have here a spot in which cells have died. And since these cells are dead and the other ones are growing, they push. The ones that are alive, which keep growing and expanding, they push. And since they push, this thing is going to produce a pattern, and this is going to produce a wrinkle. Okay? You can measure this because you can put fluorescent beads in your experiment. That's put fluorescent bead inside the biofilm, and then you follow, you track, like you do in fluid problems, you track these beads, and then you see how the machine moves, and you can see that where the cells die, the other cells converge, they move towards them. And then there is a very good correlation between the pattern of convergence in red and the pattern of death in green. So this is a correlation, but we are hypothesizing in our model that there is a causation, so that the cell death is causing the wrinkles. Well, this is the correlation pattern. So if that is the case, we should be able to make experiments in which we produce induced cell death, and this leads to the wrinkles. And this is what Morihiro did many years ago already. So if you induce cell death here by just using a wire, you stick a wire in the biofilm, and then you do a smiley face, and then eventually you get a pattern with a